And what I wanted to talk about is a little introduction to the precision medicine topic in general, why bioinformatics and precision medicine, but also to tell you briefly about what the program is going to help you with when you are joining this training program. So before I go there, our background is we're a technology transfer company, and we have a close collaboration with University of Haifa, the Tauber Bioinformatics Research Center, which is a center dedicated to the development of computational tools and algorithms, and also making them available in a easy to use way for many users that do not have an engineering and a bioinformatics background. And essentially the objective of the OmicsLogic program that we developed is to help with training in bioinformatics, to enable independent research that is guided by mentors and peer example. We want to develop a community that is thriving and growing, and they share a passion for data-driven research and an appreciation of citizen science, which means that not just scientists doing science, but people that are learning about science are also engaged in that process. And then we also want to make sure that we are enabling students, clinicians, and faculty of all backgrounds to develop novel independent research using latest technology in life and data sciences, which means to make it more accessible and more user-friendly. And so today we're speaking about precision medicine, and I want to briefly talk about why bioinformatics and precision medicine. I mean, if you're interested in precision medicine, maybe you're a clinician, maybe you're a student in biotechnology, maybe you're just interested in applications of data for the biomedical research. And so the question is, well, why bioinformatics? So what we want to talk about today is how essentially the availability of data and high throughput computational resources and computational resources that can analyze and integrate and visualize this data is actually driving a new approach to using data for clinical purposes. And so to describe what that really means, I want to take a couple of examples, and then I'll pass it on to some of our mentors to speak more in depth about their particular examples with research studies they've been involved in. So let's take an example with cancer, right? We know that cancer incidence and mortality is growing. Specifically, it is growing in countries that have already good healthcare systems. They have a lot of research. And the major driving force behind that is the growing and aging population. So the percentage of people that are older, typically that is seen as one of the driving reasons why the number of cancer patients and mortality rates are increasing. Now, how do you leverage the ability of these countries to understand and study these diseases to address such a pressing challenge that is only bound to continue and grow and become more complex in the future? Well, one of the things that is now obvious about cancer is that the earlier we understand that cancer risk is increasing, and the earlier we can detect the specific type of cancer that is developing, we can develop markers of cancer risk, and we start addressing that cancer earlier on. And that is a proven way to address cancer mortality and treatment in a more efficacious way. And so this early diagnosis really starts very early, and maybe for some cancers, including cancers that children have, this is a very critical point. So we need the ability to screen, but obviously we cannot screen for everything. We need to know what we're looking for. And so the basis of this data-driven and biomarker-driven medicine is really having access to data. And so in the past couple of decades, I think the major exciting thing that has been happening is that next generation sequencing as a technology has been refined to the point that it is becoming accessible enough and affordable enough. So it is really entering the clinical domain. And so it is entering with this diversity and challenge that a technology like next generation sequencing can bring, because essentially next generation sequencing can be used to generate a diverse set of data. And so that brings us to this point of omics data. So why omics data? And as you know, we're called OmicsLogic. So a lot of this program is going to be focused on omics data. So omics means a collection of the totality of something. And so it could really refer to a number of different things. For example, we can say phenomics, which is the collection of phenotypes, because really when we look at cells, let's say coming from a tumor or the microenvironment, they have different morphology. They have different characteristics that we can define in different ways. And so we can collectively call that phenomics, the collection of phenotypes about a particular organism, cell, or tissue. We can also generate genomic data, right? So a lot of us know about genomic data, genomic medicine, essentially looking for those mutations or finding associations between mutations and specific types of cancer or specific stage of cancer or specific treatment that might be effective or not. So that's called genomics. We're looking at mutations, changes in the DNA. Then there's epigenetics or epigenomics, a rapidly growing field, because more and more we're understanding that genomic regulation, what's written in the DNA, is also regulated by additional elements like histone modification, like DNA methylation, how chromatin is open or closed and how accessible it is, and many other factors as well. So a growing area of research that definitely has an effect on cancer as well, because we know that cancer is driven by genetics, but also by the environment. And then transcriptomics, where we look at gene expression, alternative splicing, gene fusion, and many other interactions between mRNA and the mechanisms that are a part of the translation mechanism that turns the written genes in the DNA into actual proteins that are being used. And not only is that a useful proxy to look at proteins, but an important view of what are the specific biological processes that are happening right now in a given group of cells, individual cells, tissues, organs, or the full organism. And so what is, I think, exciting about availability of this data is that it provides us with a very detailed view of not only the result, the product of what is in the DNA, but it actually gives us a snapshot of what is being done right now. So if you think about how this affects clinical use, you can imagine that if before we were looking at expressed proteins, if we were looking at histology, if we were looking at morphology of the tissue and differences in the phenotype, now we can look deeper inside every single cell, and we can actually understand what is going on there right now. So how is this changing cancer treatment? Well, one important factor is that it is actually right now changing breast cancer classification as one example. So not only are we looking at the changing practices in the clinic because of next generation sequencing data, but we're actually looking at an active area of research that makes a lot of these clinical approaches to treatment, diagnosis, and prevention of cancer a dynamic area of research. And so we're really looking at now how we can look at histological classification, functional classification, and molecular classification together by integrating multiple types of omics data through omics integration, but also then how to use machine learning on large data sets that we already have available to integrate and make sense of this data in the context of disease classification, but also for individual patients. And I think that's why really we're looking at an important and a growing role of bioinformatics and informatics and data science in general in this clinical field. So it's really about bridging the early stage basic research using this data to translate this research to practice and understand how different aspects of omics data could be applied in practical clinical situations. Now, that's one level of complexity, multiple omics, multiple types of challenges. But another one is that really we need to connect how this basic type of research really connects to what we see in patients. And typically that means how do we translate insights from patient-derived cell lines to studies that are being performed on animal models, to then the application of that information for clinical trials and also into the clinic, which is the ultimate goal of genomic medicine. So a good example of that and how it is changing not only with omics data, but with omics data as well, is this application. And here's an example where we are looking at existing types of cancers. We're looking at new types of disease states that we can identify. And ultimately, instead of approaching that from a typical clinical perspective, we're really looking at a data-driven perspective where we are trying to actually leverage the availability of data, and a lot of that data is available in many different repositories, and I'll briefly mention that, to essentially speed up the new development of treatments and clinical applications to leverage that data, treat patients better, develop new or repurpose drugs to treat disease better, and also to even prevent data by understanding better what are the driving risks behind many of these types of diseases. So as I mentioned, surprisingly, not only is this data available, the availability of data is growing at a rapid speed. And so what we see, especially in this pandemic, right, we've seen how the availability of genomic data, not only is it growing in volume, it is also growing in how fast it is becoming available, right? So if you imagine as somebody who is maybe a student or a clinician, you know, where do I get all of this data, right? So there's so much data becoming available every day that we can mine and learn from and understand. It is amazing that we can leverage such expertise and investment of others into generating high quality data sets, really generated from, like I mentioned, cell lines, animal models, as well as patients. But the problem is really that we need to learn how to navigate this data, right? So here in this image, you can see this is mapping of reads to the reference genome, right? Every single box right here represents a piece of data. So working with this data and making it useful, really extracting some meaningful insights, is challenging. And that is why we can hear about all of these exciting methods, people, you know, doing amazing projects and maybe integrating a lot of this data and running all kinds of artificial intelligence on it. And the problem is that it seems out of reach, right? It is just complex because you have to deal with huge amounts of data and very complicated methods. So how do you get trained? How do you get into something like that? Or if you're already in it, how do you expand your horizon so that you can actually leverage some of these more advanced methods and speed up the process of utilizing bioinformatics to actually ask such questions and do some kind of meaningful work in this field? And so our solution to this is to try and make tools for analysis and integration of this data more user-friendly, and to train people with the logic behind how these tools work so that anyone can play around with big data sets, analyzing and integrating them together, asking research questions that make sense and that will essentially help you understand both the data and the computational approaches in a better way. And so what I mean by that is that we built the logic of analysis into the toolkit that we have, and we'll talk briefly about some of the resources that you'll have access to in this program, where the logic is explained in courses and applied in a meaningful way in a computational big data analysis platform that you can point and click, getting quickly from raw data sets to structured data sets, developing tests and annotating them with biological information so that you can quickly go from raw data available on these public repositories to biologically meaningful results. And so this kind of annotation, I think, provides us with an opportunity to make complicated methods that ultimately are required to make sense of this data easily approachable, and we can now take examples from real studies that will allow us to ask these questions and understand what others have done. For example, availability of the Cancer Genome Atlas, or the TCGA, really is one of those examples where we can source multi-omics data from 12 different tumor types with thousands of patients that have information about their clinical stage, about the different types of tumors, about the different types of treatments that they received, and even comorbidities in many cases, and start asking meaningful questions about how that data could be used to inform the classification of patients, the treatment strategy, the prevention, the risk, right? So all of these important questions could be asked. Not only that, we will also take a look at some examples where cancer studies using patient-derived cell lines could be used to train mathematical models using machine learning and classification to start predicting different classes based on signatures or specific molecular biomarkers. We will also dive deeper into some of the clinical data for risk factors and outcomes. We'll take a look at publications in top journals like Cell and Nature Medicine that actually show us how do people utilize some of these types of data to come up with new computational methods, but also how to apply known bioinformatics approaches that you will learn in this program to extract meaningful insights. So cancer obviously is one of those examples. Another example, a type of diseases that we know is infectious disease, is also a growing area where molecular precision is becoming more and more informative. And I'll give you just a quick example. We all are hearing about these new variants coming up in different strains of coronavirus all the time. And what we know now is that these strains are defined by mutations, and those mutations have a critical impact on the rollout of vaccines, right? We actually hear about that every day in the news. And it's amazing that today genomic data is so easy to talk about, even though just 5 years ago, you would start talking about genomic mutations and people would just stop listening to you, right? So we live in an era of genomic data becoming more and more relevant to everything that we interact with. And so this application of molecular biomarkers and genomic variants to insights that have a practical public health and also clinical impact and significance is now very obvious. So this program is going to talk about some applications of computational methods to a variety of clinical biomedical research, pharma and biotechnology challenges using big data that is sourced from peer-reviewed journals and found in different repositories that anyone can get data from. And it is going to be oriented towards where this field is going. And it's obvious that this field is going towards big data, where data mining and classification methods are becoming more and more user-friendly and easy to apply to these data sets. Extracting meaningful signal from that data is becoming easier. But at the same time, what you really need is you need the logic of bridging the gap between the biomedical and the clinical applications of this data to the actual methodology so that the two can be used together in a meaningful way. And so in this program, we're going to go through a lot of these examples with some also technical, practical training that you can get access to, how to perform many of these analytical approaches on our big data analysis platform, including in R and Python, learning from curated examples and then developing a sense of a computational process where you go from raw data all the way to biologically meaningful insight, and taking that with you so that you can start developing an environment for yourself where you can easily leverage this data for meaningful biological and biomedical research. And so some of the outcomes that we envision for those that are joining this program is to understand scientific literature that describes analysis of omics data, to know how to do basic processing steps for next generation sequencing data analysis with consistent accuracy and meaningful approaches, to perform exploratory analysis and generate a hypothesis from data, to design contrast for statistical tests and prepare data for machine learning, annotate found signal using biological datasets and references, identify appropriate data sets for answering a biological question, how to plan a bioinformatics analysis from start to finish in an independent way, and then how to present a project proposal, including the rationale, the methods, scientific background, and anticipated results. And so if this is relevant for you, and I want to mention that we've previously had hundreds of people finish this program and really taken away meaningful training skills that they could later apply to a variety of challenges that they face in their own research. And so that includes many different participants of different backgrounds, including high school students that are ambitious and want to get into a program with an advanced project that they can present. Includes undergraduates that are learning something relevant and really want to get ahead and maybe want to prepare for their master's studies, or they want to go into a more data-driven field. That includes PhD students and postdocs that are now engaged in different types of data analysis projects, and they need the skill set and the tools and resources to make their understanding of that project more data-driven and apply these tools in a meaningful way. And of course, researchers and faculty that are also trying to leverage bioinformatics to make their research more impactful. So now I would like to pass it